Welcome back to the Vitrolife Group Academy studio here at ESHRE in Amsterdam. Next on our program is a discussion about the way that we incubate our embryos. There's a lot being said about incubation in terms of its humidity, dry incubation, non-humidified incubation. What is the difference and what is the impact of it on the embryos? I have with me Francesca Bahr, who's going to tell us more about that. Thank you, Francesca.
Thank you, Jaco. So I'm going to talk today about safe culture in a non-humidified environment. And there's been a lot of talk and discussions in social media lately about culture in dry and humidified incubators. And I'd like to talk a little bit about the considerations of how you safely culture in non-humidified, and then also show how some of these studies have been done, and then finally some validation in our Embryo Scope to show that we successfully can have good embryo development in a non-humidified environment. So that's the agenda. Now, when we're talking humidity and non-humidity, it's not the question of humidity. What we're really discussing is something called osmolality. And essentially, this is the concentration of molecules per weight of a solute, in this case water, and that's as opposed to osmolarity, which is by volume. We use osmolality because weight is not as susceptible to temperature changes. So you can see here 2 droplets, same number of molecules, but by the weight, the one on the left is less osmolality than the one on the right. So as we know, media droplets, they contain all kinds of nutrients to support the culture of the embryo, salts, amino acids, sugars, but they also need to be in a concentration that supports an equal tonicity of the embryo. So if your hypotonic solution, too low a concentration, then the cell begins to explode, like if you put a red blood cell in water. And if it's too high, the cells shrivel. Just like when you go on the beach and you're in the salt water too long, your fingers will start to shrivel. So what we want is a very equal, normal concentration of both to maintain the tonicity. So they have to have the right balance of molecules in and out, and there's mechanisms of transporting water that support this. The actual osmolality limit that's known to support good embryo development, the upper limit is about 300 what we call milliosmoles per kilogram, and that's what's accepted for human embryo culture. However, embryos can also use components in the media, both inorganic and organic osmolytes, such as glycine, to control the osmolality. So it's not just a simple look at the number of molecules. Yes? Again, I'd like to right away clear up, because I hear this discussion of dry and humidified incubator, but in fact, some incubators using gas mixers are not dry. They're actually proportion of the ambient humidity is included in this system. So this study was done in ESHRE 2018, and you can see in a humidified incubator you get about 80% humidity, but in some benchtop incubators you do get some humidity. In the Embryo Scope it's about 25% given an ambient lab humidity of 40 to 60. The only time you really get a dry incubation is in a benchtop incubator with a pre-mixed gas, because there's no humidity. So we need to discern between dry, non-humidified, and humidified. So there's different things that can affect these shifts in osmolality. One of them is the role of oil. As we know, clinics typically use oil overlay or humidification to control this evaporation of media to prevent these osmolality changes. So oil overlay prevents humidification, but also small droplets. It's about the droplet size. If you have droplets, there's actually a large surface area exposed that can increase the evaporation. Oil alone, however, is effective on its own without humidification to prevent osmolality shifts, provided you have a higher enough oil overlay. So this is very crucial. So the role of the culture dish here, I just showed you an example with a droplet, which a large surface area. However, in our culture dishes, you have microwells. So it's only the surface, the rest of it is protected by a dish. So this minimizes evaporation. You don't have such a large surface area. And there's been some studies looking at the role of time-lapse culture dishes in osmolality shifts. So here have been 2 independent studies looking at different dish designs and the increase in osmolality over time in non-humidified. Osmolality will increase, but the question here is how much does it increase and does it stay at a safe level? The bottom study was a recent publication by Mestris et al. At Embryo Tools, also looking at different culture dishes. And if we look at the order of methods and materials, as well as our internal validation, our embryo slide dishes are in the bottom here. So there is a slight increase in osmolality, but not sufficient to harm embryo development. Steve Mullen also did a fantastic study. I encourage you to read this paper if you're really interested in this, looking at the effect of many different things on osmolality. And this included surface area to volume of your media, oil level, oil density. There's very many calculations. And he created a calculation model that if you input these, you can get an estimate. And this is a 3D curve looking at increase in osmolality per hour. And then you look at oil height and also surface area to volume. And I've indicated here where on this curve our particular culture condition lies with stabilizing osmolality. So you can see it's very low increases. And in fact, with our real measurements, we can see it's about 0.9% change per day. So another role of looking at the safety is the starting osmolality of the culture media. So some culture medias have a very high starting osmolality, 285 to 295, that's their starting range. Whereas our GTL and GXTL have been designed specifically for extended culture in a time-lapse system, so we have a much lower starting osmolality to support this. And we also have components that the embryos can use to prevent osmotic shock, particularly some amino acids like glycine. So we see so many studies out there saying dry and non-dry, but these studies sometimes are lacking information. The type of incubator you're using, are you using truly a benchtop incubator? Are the starting osmolality of the media, what have they been using? The gas source, if you're, like I said, if it's a premix gas, that is dry, versus if it's a gas mixer incubator. And then also the method of humidification. I see a lot of these studies, they don't even report that they've actually measured humidification or for osmolality in that matter. So they just say dry, not dry. There's no measurements showing osmolality, humidity, or sometimes the osmolality is measured by weighing the dish, which is not as accurate as an actual osmometer. And again, the dish culture design, is it micro droplet? How big are those droplets? How much oil was used? So also, if you're running an incubator under humidified or non-humidified, you have to calibrate your CO2 sensors to adjust to that. So some of these studies we see there's a lot of lacking information. This is the most cited study for looking at humidified versus non-humidified. This is the FOSSI study. However, there's very many flaws with this study. First of all, humidification was achieved apparently by putting a water dish in one of the chambers. And the problem with this is an incubator type where when you close the chamber, the air is recirculated all around. So given that humidity wasn't mentioned, it's very impossible to see if this was actually humidified because the air is circulating all around. Neither humidification levels nor osmolality were actually shown at all in these. Furthermore, it was a small microdroplet, 15 microliters with 3 embryos in it. So this would exasperate us, particularly if you're looking at these systems. So this study cannot really conclude that all non-humidified incubation is detrimental by any stretch. However, it is the most cited. So I'm going to look at validation studies in our system in terms of osmolality. This was a GTL and olive oil heavy, and we were looking at start and day 6, the increase in osmolality. You can see here, yeah, there's a slight risk, but that red line is what is the accepted safety limit. We do not come anywhere close to this even after 6 days. And what you will see is, like, for example, the GEMS media, we don't even end where their culture media osmolality starts. So, yeah, we rise a little bit in osmolality, but is it unsafe? No, absolutely not. This is another study. This is not from our in-house data. This is another external data that I've seen shown, again, with a humidified and Embryo Scope. Again, we see almost exactly the same ending osmolality, about 280, even after 6 days of culture. So we know we're very, very safe here. Media composition also plays a role. We've just done some recent studies with our GTL and GXTL, and you can see even with the GXTL and the GTL, the raise in osmolality is different depending on the components of the media. So this is another consideration to look at when you look at these studies. Yes? One more study, this was shown at PCRS, and it was also a MIA assay. So cells were counted after 6 days. This study is done in Denver, which is very dry. So the rise is a little bit higher, but still very safe. Exclusive to us, every single instrument we release goes through a MIA test. We are the only time-lapse instrument that does a MIA test. And that green one, no, you cannot buy a green embryoscope, unfortunately. That's called our reference embryoscope. And on the left, you see all our instruments lining up ready for their MIA test. And we were running a MIA test last week, just before I left. You normally stop at 96 hours, but I said, let's just keep running those mice. And this is at 112 hours, plus an 18-hour of dish equilibration. These blastocysts had been hatched since 72 hours, just growing fine. So raises in osmolality are not detrimental to embryo quality necessarily if they're not too high. But the real truth in humidified, non-humidified is looking at the embryo development. And if you're not reporting actual embryo development, you're not learning anything. So you can't just say dry, non-humidified. And we have very many studies showing excellent embryo culture in our system with our dish type, our oil level, and comparing to both humidified and non-humidified incubators. And in here in red, I've pointed data. You can see usable blastocyst rate, number of blastocysts. Our system supports good embryo development despite the fact that it's non-humidified. So safe culture in the embryo scope, these are some considerations, and people are trained on this. We get a full training session when we install. Use media design for extended time-lapse culture. Use a GTL or a GTXL with the TL in it. It's designed for time-lapse. Use sufficient oil. And this also means that when you're pipetting the oil, you need to be careful that you've expelled the full amount. If you've got a heavy oil, sometimes some of it stays in. So maybe load with a little bit more oil. Be fast with your dish prep. If you're not using your rinsing wells and putting media, you should also account for that in your oil. And then, consider a little bit. You can put up to 1.8 milliliters easily in our dish. And you can visit our blog to see we have a blog post about how to maintain stable culture. And summary. Studies support that non-humidified incubation under the right conditions is perfectly safe, and you can have excellent culture. Even though the osmolality raises, it does not reach these critical levels that might affect embryo development. And use a culture media that's designed to support extended culture over multiple days. And then the added bonus of why we would want to culture without humidity is simply the contamination issues go all away. You don't need downtime for cleaning, and the risk, I don't think we've ever had fungal or microbial contamination. So this is a huge bonus in non-humidified culture. So I hope I've all convinced you that non-humidified culture is not bad and dry good. It's a helicopter view. You have to look at the whole culture system and how it supports it. And that's all I have, and I'm happy to answer any questions. Thank you.
Thank you, Francesca. Very, very interesting there. And it's good that we understand more about the difference between dry incubation and non-humidified incubation.
Exactly.
There's a big difference. I think one of the points you raised, which is also very important, is that the way that we prepare dishes, especially whether you use time-lapse or not, if you're going to do extended culture without interruption, it's vital that these dishes are prepared correctly, that you don't lose osmolality before you even start the culture.
Yes, very important. Yes.
Any other questions from the floor? Okay, well, thank you. We conclude that session. Thank you. Bye-bye.
Thank you.